Hey guys, Mike Verkest from the Second Shift Podcast here to talk to you about Piace. Back in 1996, I had 2 certifications to manage, my state license and national registry. That was it. Fast forward 20-some years later, and what I would give to have just 2. Between my advanced certifications, ACLS, PALS, PHLS, you name it, I'm looking at upwards of 10 certifications to manage. That's where Piace saves my bacon. I simply add my certs, expiration dates, and bam, no more surprises. Take pictures front and back and share those credentials with whoever needs them. With the touch of a button, it's easy. Go to the App Store now for your device and search for Piace, P-I-A-C-E-T, Piace. Do it today.
Hey everybody, Eric back with you. In today's podcast, we're going to do a crossover episode. I thought this podcast was exceptional. Please join me in welcoming the Second Shift and Lighthouse Project Podcast crew and their guest physician, where they really dive deep into everything related to COVID-19, the actual SARS coronavirus, and the mRNA vaccines. They dive into all of the tough topics that we have seen in mainstream news, and I think they did an exceptional job. If you have questions related to the virus, the vaccines, I think to me it answers a lot of great questions. So please join me again for part 1 of 2, and let's get started.
The content of this podcast is based on medical fact and evidence-based practice from credible authoritative sources, but is not a substitute for your institution's policies, procedures, common sense, or good judgment. The views and opinions are those of Eric Bauer and FlightBridgeED in their entirety. This is the FlightBridgeED Podcast, critical care and emergency medicine education for nurses and paramedics. Here's your host, Eric Bauer.
Well, hey everybody, Mike Verkest joining you guys live. Thanks for jumping on with a special edition of what was intended to be an EMS Lighthouse Project podcast crossover with the Second Shift. But as you can tell by looking at your screen, we are conspicuously missing somebody, aren't we, Ritu?
Jeff Jarvis, where are you?
Yeah, I don't know. This whole thing is this thing has been hilarious, as usual, and I don't understand it, so that's okay.
Technology.
Yeah, it's just one of those things.
Yeah.
Well, here's the thing, Jarvis. I don't know. I'm assuming he's going to jump on. We'll keep an eye out for him here in a little bit, but I know that we all want to extend a special thank you, guys, for jumping on and joining us again for really what is sort of part 2 of the Second Shift, episode 81, when we talked a lot about mRNA vaccines. We talked about, everything from the beginning, right? How these things were developed, all that kind of stuff. And what our intention is today, and we'll get through this and then we'll do some introductions so you know who you're dealing with here. Most of you guys know the long-haired guy. And some of you might remember the other guy down at the bottom there. But really what we wanted to do today was just revisit. There's so much misinformation going on out there. And really it's our intention tonight for the next hour or 2. I don't know how long it's going to take. But what we want to do is be able to answer questions for you and explain things as a trusted source, because I feel like that's kind of what we are. It's not CNN. It's not Fox News. There's no political side steps. This is just people that care about you, care about our community and our families, and we just want to have straight talk, right? That seem reasonable to you guys? So that's kind of what our plan is over the next couple hours. Again, sans Jarvis, with Jarvis, I don't know. It just depends how it's going to go. But one of the things I wanted to do real quick was just do a quick introduction because there might be some of you that are watching that maybe haven't tuned into us before. So just a little bit about us. I'll start. My name is Mike Verkest. I'm a fire-based EMS training officer and just all-around EMS goob. And to be honest with you, tonight I'm just the eye candy because it's these other guys down here. And it's not the first time I've been called that, so I'm cool with it. It's totally fine. But I'm really here to kind of host the show, kind of keep things moving around a little bit because, again, we've got a lot of stuff to talk about. So with that, I will send it over to my good friend and colleague, Dr. Ritu Sani. Say hello to the people, good sir.
Hello, people. Unfortunately, we're not on our usual broadcast platform either. So I'm wondering if we'll have much viewership. But if you're listening to the podcast or watching on YouTube, that's great also. But my name is Ritu Sani, and co-host of the Second Shift podcast with my good friend Mike Fricast. I am an EMS physician and an emergency physician. I'm medical director of 2 different counties here in the Portland, Oregon area. I'm out in the suburbs of Portland. I'm not in the main part. And I work clinically as an emergency physician also, and been co-host with Mike, I don't know,
A couple weeks.
Years. Yeah. And it is not the first time that Mike has been called the eye candy. That is true.
Thank you.
But then, it's great to have back on Second Shift, Dr. Hayden Smith.
Good to be back.
Yeah, you barely squeaked through your rotation last month in the emergency department. I didn't get my evaluation turned in time, so I guess they passed you. But give us a little background on Dr. Smith.
Well, first off, I totally enjoyed working with you in the emergency department. It was a lot of fun to see you in there, see you in your element. So I am Dr. Hayden Smith, a resident here in Portland at Providence Portland Medical Center. I was on this broadcast previously talking about mRNA vaccines, and with a lot of the information that's online, with a lot of the changes that's been going on as far as the science around the vaccine, I thought I'd come back on and talk a little bit more about the science of the vaccine, what we now know, and answer some questions.
Well, isn't that perfect? Because that's exactly what we want to do. And one of the things that I have done over the last, day or two. So for those of you that are regular Second Shift kind of watchers, you know that generally we don't put a lot of planning into what's going on, right? We're pretty good at, we're just going to go with it, and however it goes, it goes. But this one we actually put a little thought into, right? Because we wanted to make sure that, for this, we even got dressed up. Look at Ritu. He's in like a polo shirt. You don't ever see that.
FlightBridge polo.
I know. It's pretty good.
You guys are matching.
I know. We do match.
Yeah.
That is the sweetest
thing.
People
so often can confuse us for twins.
Constantly. Well, one of the things that we wanted to start off with was really going sort of back to basics, right? And Hayden, what we thought we could do was start off with you and talk us a little bit about what is a virus.
Yeah, I'd love to. And I just wanted to second what Mike was saying, that, these guys have put a lot into trying to answer your questions and understand the facts behind what is out there. And so, there's a lot of good stuff that we want to talk about today. And so, we're really excited to talk about it and answer those questions. But we do want to get back to the basics to start out with and talk about what is a virus, because I think it's important to understand the basis of what is going on, kind of start from scratch, so that we can then kind of build and talk about some of the more nuanced topics with the mRNA vaccine and COVID and things like that. So, simply speaking, a virus is a small particle, a small kind of molecule, almost very complex molecule made up of different proteins that is not really living, but what it does, because it can't replicate itself. And so what a virus has to do is it has to get inside of a cell in order to hijack some of the materials that are in the cell in order to replicate and grow. So a virus is just some particles of protein and some DNA or RNA. And there's different kinds of viruses that use both DNA and RNA. And when this viral particle gets inside of the cell, it steals that machinery from the cell, and then it uses that to replicate and create more and more particles until the cell gets so filled with particles that it essentially explodes, releasing all the particles out into the serum. And then those viral particles can then go and replicate in other cells. So it's just this kind of cascade of worsening infection as it starts in one cell, then thousands, then hundreds of thousands, and then millions of cells. As this virus grows really fast and really rapidly, it makes it a really good infection, to be quite honest. And so our body has ways of attacking that. It has ways of combating viruses. It's been doing it for hundreds of thousands of years. And so as we have evolved to combat viruses, our immune system attacks the viruses in two different ways. You have antibodies that go and recognize and attack specific viruses. And then you have a different kind of cellular immune response that goes and attacks the cells that are making the viruses. And so on one hand, your body can sequester or, kind of stop the viruses with the antibodies, and then it marks it. And that's the important thing, is it marks it with the antibodies to then go and attack, eat it, and destroy it. Now, another mark is put onto the cells that are making the viruses. And the body goes in and then essentially kills the cells that have the virus inside of it so that it can stop it at the source. And the body sees that as a benefit to just killing its cells that are infected, because it has, cells that can take the place. And, if you don't do that, then then the virus goes unchecked and just grows, proliferates and infects other cells. So that's kind of how your body reacts to viruses, as you have this antibody immune response and this cell immune response. So kind of moving on to COVID. COVID is a specific kind of virus. It's actually an RNA virus. So you have DNA and you have RNA. DNA is permanent. That's what we think of when we think of genetic material. RNA is like the temporary template that that your body uses to make proteins, but then it goes away really quickly. As a result, though, because COVID uses RNA, it means that it can get translated really quickly. And so that means that it can infect a lot of cells a lot quicker. It doesn't have to go through the step of DNA.
So I've often heard that people refer to the RNA as like a blueprint, right? Like these are the plans, right? Go forth and do your thing. Is that accurate?
Exactly. Yeah, that's very accurate. So it's like the blueprint that the cell then uses to create proteins. So then,
I like the DNA is the blueprint, and like the RNA is like a copy of the blueprint that you give. Like the plumber dude comes by, and he's like, "I need the plumbing blueprint," and you give him just that copy. So the RNA is a copy of the protein that it's being made right now, and it goes out in the cell and makes just that protein.
Yeah, you can think of the DNA as kind of like the master plans, and the RNA is like a specific instruction.
Okay.
So, here's your instructions, go and do this. And then the DNA holds all of the information. And so once you have this RNA, you can create the proteins, and the virus takes advantage of this. And COVID is a coronavirus that has this RNA that then invades into the cells. And it invades by using two proteins. One is a viral protein, and another is a human protein. The viral protein you've heard all the time. It's called spike protein. And the spike protein attaches itself into the ACE2 receptor on your cells. So when that happens, they connect, and then the virus binds to your cell, and then it gets the signal, your cell gets the signal to bring that viral particle into the body, into the cell rather, and then the coronavirus then releases the RNA to infect the cell and cause the damage we were talking about before. And so so that's how it gets in. And that spike protein and the ACE2 receptor are proteins that we'll probably talk about here during the podcast, which is why I bring them up. And then another point, probably I'll kind of leave it to the guys after, but I think one other point I want to make is kind kind of just a naming thing. So COVID is what we hear all the time, but that's the infection. That's the syndrome. You think of like, another name or another syndrome that's commonly used is like Down syndrome. It's a collection of symptoms that we then see and call COVID. The actual virus is called SARS-CoV-2, and it's called a coronavirus. So they're actually two different things. We use them pretty interchangeably, but that is the difference between those two names.
Okay. That makes a lot of sense to me. But coronaviruses aren't new, right? I mean, they've been around forever, right?
Yeah, no, they're not new. There's a lot of different kinds of coronaviruses. Coronavirus gets its name from the name crown. Corona means crown, and it's directly referencing those spike proteins that are that are kind of around the virus. And they've been around forever. They've been around for a long time. SARS, the first SARS is an infection that was in China, and then MERS was the first coronavirus that made. I believe it was the first coronavirus that made kind of news and headlines.
Yeah, the first SARS actually hit Toronto really hard. So that was for those of us who were around then and have a lot of EMS friends who work in Toronto. They and that, Hayden, is an interesting piece of history, because if you go back and read the Toronto experience with SARS, with the first SARS,
yeah,
it's like this experience but shrunk down.
Mm-hmm.
They had a lot of similar issues around like the super spreader events. So the original COVID, it was very contagious, but it really wasn't equally contagious. It was sort of like some people were really a lot more contagious and other people weren't at all. And one of the super spreaders, I think, happened to be a paramedic. And at the time and actually went to different facilities is my recollection. But there were a lot of the same challenges around like how much do we close down, how much, that sort of thing. So that was what I can't even remember SARS when that was, early 2000s. So when Hayden was like 6 or something like that, or 5.
Time flies.
So, I mean, let's jump forward a little bit. So let's just, for all intents and purposes, we'll just use January of 2020. There's this new thing coming around. We're starting to see it. Wuhan, China, big deal. A lot of cases coming out of there. Then we start seeing the spread. And next thing you know, SARS-CoV-2, what we see now, we start seeing people with COVID disease. And then let's fast forward through 11 months-ish, right? And bring us to about December of 2020. And all of a sudden, I'm using sort of air quotes there a little bit, we start hearing that there's been some vaccines that have been sort of being developed really since the summertime, maybe even a little bit sooner than that. And the question becomes, well, what is this vaccine? What is this technology? What is mRNA? And we start hearing all this stuff. Oh my gosh, it's so brand new and all that kind of stuff. And so maybe you could talk a little bit about what is an mRNA vaccine, such as the Pfizer and the Moderna specifically. And how do those work exactly?
Yeah, great question. And it's one that I get asked all the time. Like friends, family, they always ask me, what are these vaccines? What's this mRNA that they're talking about? So I will first start with, the term mRNA. So mRNA is those instructions I was talking about. When your body naturally creates proteins, it takes a DNA and turns that into RNA, an mRNA molecule that then goes out of the nucleus and then gets transcribed and makes proteins. So mRNA, it's called messenger RNA, and it's kind of the working, the template that they use to make the proteins. So that's mRNA. The idea to put mRNA into a vaccine is not a new one. It actually harkens back to, I believe, a patent or the idea goes back to 1989, I believe. Just for reference, that was before I was born. And it was,
I was in college.
And so the idea behind this, rudimentary mRNA vaccine back in the early 90s was to inject, just straight raw mRNA into the body to see if it creates an immune response. Because the thought is that if you can put this mRNA into the cells, then they can then create the protein, and you can select what kind of proteins you create an immune response to. And so they did that, and it really didn't have any of effect. It actually caused a ton of side effects, and I think only one person ever created antibodies to this naked mRNA that was injected through these rudimentary mRNA vaccines. Nothing really came of it. It wasn't until 1993 that they started working with kind of protecting the mRNA in what's called a lipid. It's called a micelle, but you think of it like a fat envelope. So it's kind of you put the mRNA inside of a little package.
Dr. Jarvis refers to it as a tiny drop of butter.
Yes. Exactly.
Which, if he would have got himself logged on, he could have told us.
He would have said, "Howdy, y'all! It's a tiny drop of butter,
and it's a little drop of butter."
It's a little
exactly
drop of
what
butter.
It is. So they started putting this mRNA inside of the tiny drops of butter, and that allowed them to get the mRNA into the cells. And then companies started popping up that started using this technology to really start to get serious about this research and start making vaccines. And so that's where two companies that you've heard of a lot, Moderna and BioNTech Pfizer. And I'll use Moderna and BioNTech because those are the two ones that actually produce the mRNA vaccine.
Right.
And so Moderna and BioNTech, they started doing this in cancer and infections in a lot of different areas to try and create this immune response. And that harkens back clear to 2000, believe it was 2008 was when BioNTech was made, and 2010 was when Moderna was started. So this technology is pretty old. If you're taking Moderna, for instance, that was 10 years before the mRNA vaccine that we have today. And so we have lots of data from clinical trials, from old studies, and things that go back to 30 years ago. I mean, the research goes back to 1990, 1989, 1990, when these first studies were being done. And so the thought that this is a new technology doesn't hold too much water, because we do have a lot of data and a lot of safety data with these vaccines.
So I want to make sure that, I think we covered some really important things there, and I just want to make sure that let's recap this last piece here. All right.
Yeah, great.
And I just want to, and I'm a dumb guy, and I just want to make sure I understand it, and I want to make sure that people that are watching that maybe don't have really a background in any kind of medical stuff at all, I just want to make sure that it's clear to them. So when you go and get Pfizer, Moderna, well, then again, we're just speaking specifically to these two. And hello, Dr. Jarvis.
Say it.
Hello.
No, he didn't say it.
Can you hear me?
Say it.
Yeah, we can hear you, but we were hoping you were going to say,
howdy, y'all. This is Dr. Jeff Jarvis.
Thank you very much. All right. Sorry. So again, if you're new to kind of watching us, we have these little squirrel moments, so it's okay. I'm just glad to see him on here. But let's recap. So I go in and get a vaccine. Essentially, what's happening is there is instructions in the little drops of butter that get injected into my arm, and that goes to our cells, and and please correct me if I'm wrong, but I'm just trying to keep it simple for myself. And it goes to those cells and provides instructions to those cells via the mRNA, which is called messenger RNA. And it says, be on the lookout for these things, right? And then it creates. So this is where I just want to make sure that we're getting it clear, because there's a lot of people are thinking it messes with your DNA and all that stuff. It doesn't. It's simply providing your cells with instructions saying, this is the thing I want you to look out for. It's got these things on it, and like that's what you want to watch for. Is that sort of close?
That's exactly right. So the vaccine essentially gets this mRNA into your cells, and then it's more of a tag than it is anything else. So it essentially tags that cell and says, here, I have something new. And in the case of COVID, that's spike protein. It says, here, I have something new. Can you mount an immune response to this? Because I don't like it. I want to get rid of it. And so that's the message that is encoded in the mRNA. Doesn't get anywhere near the nucleus. And I think that's an important point I want to make is these mRNA vaccines, this mRNA, it's impossible for it to get into the nucleus. It's kind of a one-way street out of the nucleus for mRNA. So it can't get in.
Yeah. I think a couple other. So my understanding, Dr. Smith, is that this mRNA actually causes the cell's protein making factory to go, oh, cool, here's a new set of instructions, and it makes an actual spike protein in your cell. So it makes a spike protein, and what it does, it says, well, here's the instructions. Everybody can see I've got the instructions up. All right, we're going to make the spike protein. This is actually my fantasy football draft, but same thing. And then it says, here's a spike. And then your body goes, whoa, we don't like that spike. But the other piece that I get asked a lot about is, but that mRNA, I don't want that. But what that factory does, it goes, I made your spike protein. I'm done with these instructions. I'm just going to rip them up and throw them away.
Yeah.
And that mRNA that mRNA that went into your cell and made this spike protein is gone right after this copy of the spike protein is made.
Yeah, it's like hours and then it's used up.
Right.
So, usually your body just has to kind of keep sending out these instructions in a normal, healthy cell that's making a normal protein. But because your body actually tears up the instructions and gets rid of it just on a normal basis. That's just what it does with mRNA. So yeah, exactly. It just tears up the mRNA after, I don't think it's longer than a few hours, and then it's gone.
That makes a spike.
So, as you guys saw, we had Dr. Jeff Jarvis finally be able to jump on. Howdy, Dr. Jeff Jarvis. And I don't know
Mike.
Oh, there he is. Hello. How are you doing?
Sorry about that. Complete jury rig system here. So it's good to see you guys.
That's the calm Dr. Jeff Jarvis. Calm on the outside, raging in the middle.
Yeah, well, I
Raging.
got the text that proved that it isn't calm on the inside. So
Kind of like to baked Alaska.
That's all right. Real quick, can you introduce yourself to the people that are watching that maybe haven't joined us before?
You bet, guys. I'm Jeff Jarvis. I'm an EMS medical director and emergency physician in Central Texas.
Oh, and COVID is over there, right?
Completely.
That's what I thought.
We have the governor's executive order that says it.
Sorry, I couldn't help myself. We're no better off in Oregon. Anyway.
Hey, Mike, thank you again for asking. You ask every day if my lights are still on, and as of today, they're still on. So I appreciate that.
That's good. I'm glad to hear that. All right, let's get back on track here. Did you have anything to add to that, Dr. Jeff Jarvis, that last little discussion we were having?
Really, I thought Hayden did a great job of summarizing that. Ritu, it's actually bacon, though. It's the mRNA is wrapped in bacon.
thanks. No, I appreciate that.
They're
trying to help.
There could be a
That worked
benefit
for me.
to that.
Well, since, you know, the hesitancy seems to be in the South, we figured if we wrapped it in bacon, that might actually help a little bit. But I don't know. Maybe we just need to let more people know it's wrapped in bacon, and that would help.
That'll work.
All right. So we know we've sort of talked about what is a virus. We talked about specifically what is COVID, what is SARS-CoV-2, which creates the disease process known as COVID, right? That we talked about.
Yeah.
We talked a little bit about the sort of the vaccine, sort of how it's developed. But one of the questions that people ask a lot, and I hear about it a lot, I think we all do, and that is, how did this get approved so fast? And we'll talk about what an EUA is here in a minute, but really, Dr. Sani, I'll throw this one to you first. People don't know if there's going to be a new medication or a new vaccine or something like that comes to the market. There are a number of steps and a number of processes that obviously have to be gone through for that medication or the vaccine or whatever to be approved. And there are probably a number of limitations to how something gets approved. Those things can be money, it can be time, it can be the correct kind of medication that you're looking for. I know in the past we've talked about it a little bit, and I don't want to steal your thunder too much, but if you have an ingrown toenail medication, that would probably take longer because not a lot of people have ingrown toenails, and maybe that's a bad example, but maybe just talk us through a little bit about what's sort of normal when it comes to approving something.
Yeah. Well, I think that, that I hear like, well, they invented this so quickly, and how did they get it done so quickly? So number one, Dr. Smith Hayden gave us a nice background that this is not new technology. So this idea that they made something up out of the blue and got it approved in 12 minutes isn't exactly true.
Yeah.
It's actually been around for a while, and there are a number of animal studies, and there are some small human trials on mRNA vaccines, kind of generically. But so, when this started happening, I started figuring out, well, let's look at like other vaccines, like how many patients does it take to get a vaccine approved and all those sorts of things. And one of the things that it comes down to is you need money, you need people, and you need time, right? And what was different, what was different about this vaccine is that there was a ton of money thrown at it, and there were a ton of volunteers stepping up to be part of the answer. And so when you look at something like the shingles vaccine, which had roughly 24,000 people in their trial when it was approved, it took them 4 years or 5 years to do that. And that's because you put a sign out and you say, I want people to come be in my shingles vaccine trial. You don't have like a big rush of people, there's not a daily news brief about shingles, right? Mm-hmm. And so it just takes time to enroll people in a trial for something that like that. The other thing is it takes money. And so to each person you enroll in a trial costs money. And so what you want to do is spread that money out. You're not going to get a ton of volunteers. That it's going to take a few years. Let's spread that money out over 4 years to make it kind of more cost effective. And so you have the same number of volunteers. It just takes 4 years of time. So now if you pull all that in and you look at where we were last year, people were screaming for a vaccine. There was technology out there that already exists. There was money funding billions of dollars that the government set aside to pay for this. It was pretty easy to take all of that multiple years of effort and put all that human capital, human power, cash, and people who are interested in being in the trial kind of up front. Okay. So when you look at like the COVID, like this Pfizer BioNTech, I think their trial was 35,000 people. So is that correct, guys? You guys, does that sound about right?
Sounds about. I'd have to look it up, but that does sound about right. Yeah.
Their initial trial was like 35,000 people, but they were able to enroll, and I think Jeff's looking to see exactly. They were able to enroll all of their people in that first month, right? So instead of having to spread it out for 4 to 6 years, and then they needed to get 6 months worth of 6 months worth of data, and they were able to do that within 6 or 7 months of starting the trial. And that's how they were able to sort of start a trial in sort of June of 2020 and have enough data to apply for an EUA. And we'll talk about that in a minute, as early as end of November, early December. Jeff looks like has an answer.
43,000. 43,448. There were 21,720 and 21,728 in the placebo group. So 21,000 people in each group.
So the first thing that I say to people this was not studied correctly is that there were actually more people in just the Pfizer trial than there were in any trial up to then in sort of initial trial for vaccines that led to an approval. And then if you throw in the Moderna trial, which was roughly the same size, now you have double. So they were able to condense everything because there was will of the people, there were volunteers, and there were there was enough money to pay for it. Go ahead.
And I think one thing that I also get questions about is also the science and how they kind of got that vaccine so quickly, because they're working on these other projects for years leading up to COVID. But then how are they able to develop a vaccine that they can put into trial so quickly? That's another concern that people have. And the reality is that these mRNA vaccines are very easy to manipulate and change, because all you're doing is you're changing the mRNA sequence. So really early on, I think before even 2021 hit, I think before 2020 hit, so before January 2020, we had the entire SARS-CoV-2 genome sequenced. I mean, that science is really easy to do. Right. So all they had to do was to take that sequence for the spike protein, throw it into their little butter droplets, and boom, you have a new vaccine. And so it was really easy, and something they had done for other infections like Zika and influenza before COVID. But once COVID hit, they're like, okay, we got something. We have a way we can do this. Let's put it in a vaccine and see if it works.
Yeah, it's so
the folks who were designing it, really, they've been working so long. If you think about this, I think about it as there's a delivery device and then there's a payload. So the bacon, the surrounding nanolipoparticle, that's the thing that is so hard to build. That's what took a while to build. That was built, and they were waiting for the right virus as a proof of concept to show that it worked. They thought Zika was going to be the thing.
Yeah, they did.
This is the one that's going to be able to prove that this works. The problem is Zika sort of, thank God, fizzled out. There just weren't enough cases of Zika. And then this came along. And Hayden, you're exactly right. Within 3 months of the first case in China, we had the entire genome sequenced. 1 month after that, all they had to do was take the payload and put it into the bacon. And they had that payload ready within a month.
Yeah, it was really quick.
And this is another thing where we learn how young Hayden is, because the fact that they could have the entire genome of a virus done so quickly, pretty freaking amazing.
It's a big deal.
Pretty amazing.
And we were, thinking back to when they could get the full genome of stuff done, it was pretty impressive. So
yeah. I mean, I have a background in bioinformatics and genomics and stuff like that. So, to see that happen so quickly, it was so cool to see all the genomics and stuff get done so quickly.
Yeah.
Well, and the other thing, I'm glad you brought up Zika, because that really is the vector that Moderna tried to get this mRNA vaccine thing going. And if you think about the patients that have Zika, it's pregnant patients. And so, they were kind of working to get it both safe in pregnant people. So it had to be a really safe vaccine, and it had to be effective against Zika. And so, that is another testament to how the safety of these vaccines really is baked into their creation, way before COVID came on.
Yeah.
So here's the thing that is getting me so excited about this technology. First off, I'm really glad that we have a vaccine that is as effective as it is against COVID. But I look at the concept of this. The concept is you have this delivery device, you just load in a new payload and off it goes. The ability to create rapidly vaccines, not just against viruses, but against problems, say, with proteins like cystic fibrosis. This technology has amazing potential. So I'm really excited about it.
Yeah, there's a lot of potential for it. I think, we could talk about that for hours on end. And I think we want to focus on COVID here, but yeah, there's a lot of implications in infections and cancer with mRNA vaccines. They're not going away.
So Ritu, one other thing you mentioned, you were talking about the numbers of people that were enrolled in each arm of the Pfizer trial and the Moderna trial. Let's just take the Pfizer trial, for example, 21,000 people in each arm. And yet it seems like the Pfizer vaccine is controversial. There is another product that Pfizer has out there that went through exactly the same steps with about 1,000 people in each arm. And I have yet to hear a person, certainly a single man, complain about the science behind Viagra. No complaints about that. That was 1,000 people in each arm, 21,000 people.
Right. That's exactly right. That the size of vaccine trials compared to other new medication trials in general is larger. And then when you throw in the fact that there were concurrent mRNA trials plus the J&J trial, which is a completely different technology. When you throw all that in, and then there were other trials and other products. There were hundreds of thousands of people enrolled in the trials at the same time. So the other question that I think that was there another question that you wanted to piggyback around EUAs or something like that?
I'll tell you what, before you get to the EUA, Ritu, can I just talk a little basic science here, like statistics?
Sure.
Because we frequently think that a study with a bigger N, meaning a larger sample size, is by definition a better study. The reason that the size of your study matters is in terms of power to detect things. So we could have run this study if we were looking for the power to detect a change with a couple of hundred people in each arm, because the difference that you see between vaccinated and unvaccinated is huge. So you don't need massive numbers to detect that because it's a huge thing. So the only reason you need massive numbers of people in a study is to detect things that might be really, really, really small differences. They did these massive numbers for safety because they wanted to have the statistical power to identify even little bitty differences.
Yeah,
so
I think
those numbers are there because of safety.
Yeah, that's absolutely right. And I think that, you're comparing vaccine trials to treatment trials. And they are a little bit different as far as some of the regulations go. So
but I couldn't pass up the opportunity to use the Viagra joke.
Yeah. So for vaccine trials, they have it's required to have more people in the trial because there's safety. You're giving this to a healthy person instead of giving it to a sick person, and so there's different things and different safety data that they want to collect with that. And they want to see those rare side effects that may come from a vaccine that you're giving to an otherwise healthy person. And what is great about the vaccines is they, hit the requirements that the CDC and FDA have for their trials. They just hit it really quickly because, again, there was tons of people that wanted the vaccine. And so they didn't skimp on any of the requirements for a full FDA approval. They were just able to get the numbers really fast because they had money and funding. So it's not like that the trials, they actually exceeded the expectations. If I recall, and this is, I could be a little fuzzy on the details, but the requirements that they had going into the trials was to get a 70% efficacy and have 35,000 participants. I think that's where that 35,000 comes from. And
I think
so
it was 50% efficacy, actually.
Was it 50%?
Yeah, it was. It was 50%.
And for most vaccines, that is, that's pretty okay, to be honest with you. Most vaccines have efficacies around 70%. So for this mRNA vaccine to have 95% efficacy, I mean, those are numbers that you just don't see in any other vaccine anywhere. And so it shows that, this vaccine is both safe and extremely effective.
So I think this is a good time to bring up exactly what an emergency use authorization is, because that might be a term that sort of prior to a COVID vaccine might have sounded crazy to some people or something that they have never heard of. So, Dr. Sani, what exactly is an emergency use authorization versus just something being approved?
Yeah. We'll try to make this quick because we still have a lot to cover, and it's
we do.
But it's exactly like it sounds. It was there was an emergency situation, so they wanted to authorize it. And what an EUA does, it does a few things. One, it gets you to the front of the line. So the FDA has thousands of drugs in its pipeline for approval. And the reality is that it just takes time for the bureaucracy to roll. Okay. So when you apply for an EUA, you're saying this is an emergency for whatever reason, and we want to get to the front of the line. So that's the first thing. It gets you to the front of the line. The other piece is, we've just hit full FDA approval in the last couple of weeks for the Pfizer vaccine, but FDA approval of the vaccine is not just about safety and efficacy. And that's the key. The emergency use authorization by law has to take into account the safety and the efficacy of the vaccine or drug or device that you're trying to get an EUA for. It is, and the bar for those for safety and efficacy is not changed. So they didn't look at the Pfizer vaccine or the Moderna vaccine and say, well, our normal bar for safety in a vaccine would be this high, but because it's an EUA, we're going to go like skimpy. No, the bars for safety and effectiveness or efficacy have to be the same. What is different from what makes an EUA faster is that there are a number of other things that go into an FDA approval besides just the safety and efficacy of the drug. Manufacturing processes, those sorts of things. That's number one. And number two, to be fair and honest, the standard for a vaccine trial is not only 6 months worth of data, safety and efficacy, but a full year of safety data. And so they actually applied for the EUA before they had a full year of data. So they weren't even allowed to apply until they had one full year of data. And so somebody's going to go, aha, well, they skimped on safety. But the reality is that every other vaccine trial ever done showed that if there was a problem with the vaccine, it was within the first 2 weeks. And so that was their justification for saying they could apply for an EUA after 6 months. And so they applied for that emergency use authorization that they then obtained, and then went on. Pfizer has gone on to obtain that for down to age 12. And then now received full FDA authorization, meaning they have 1 full year of data, meaning that they had the FDA came to the factories and actually did a top to bottom scrubbing of, reviewing every single process in the factory. It's very time consuming to do all this sort of work. And they, to get the full FDA approval. And so that's why they ended up, that's the difference between an EUA and a full FDA approval. But there was no skimping on safety to get to where we were. And again, the efficacy was much better than the goal, which was somewhere between 50 and 70%.
Hey, Ritu, one thing that I'd like to point out here, because I've heard it, I think, confabulated. So a lot of people will say they don't want to take the vaccine because it's experimental. They think the E in EUA is for experimental.
Correct. It's not. People think that's the E is for emergency. And the other piece is that the trial for the trial was not enrolling any more people. So no, nobody who got the vaccine once it got EUA was part of the trial, except for the monitoring phase. So every vaccine, once it's approved, there has to be basically continuous monitoring. So that's in the trial plan but the reality is that never goes away. But the E stands for emergency use authorization, not experimental. You're not part of the experiment if you got it after the EUA. Only the folks who signed up, gave informed consent, and were enrolled in, as early as June, I think, of 2020.
Like my daughter, by the way. My daughter was in the Moderna trial.
Oh, was she?
We were pretty sure that she got the active arm. She got a fever, and I was like, I don't think a placebo is going to give you a fever.
There were people in the placebo arm that got a fever, though.
Probably so.
There were, because some percentage of those people are going to get sick, get a fever within a week anyway.
Well, and just because it's a placebo doesn't mean it's not you don't have real things. Placebos are strong things.
This has been a production of the FlightBridgeED Podcast, leading the way in pre-hospital critical care and emergency medicine education.